Hello students, now we are going to discuss about the biotechnology techniques, right? So different techniques which is involved into the field of biotechnology that we are going to discuss into the different fields, say for example in bioreactors, right? In earlier times the bioreactors were used for the centuries to make the wine and beer, right? So different type bioreactor means it is the small big vessels where we are maintaining the proper pH and temperature to product to produce the particular types of products, say for example wine and beer. So that was used up before also. So the bioreactor is the possibly to most important single piece of equipment used into the biotechnology, right? That's what single piece of equipment which will be having like a bucket where we can put the cap, and we will maintain the optimum temperature, optimum pH and all those things. Bioreactors are the container or vessel that allow the biological process to take place under the optimum condition, right? That's what optimum condition means, temperature, pH and all other pressure and other thing, right? So it is nothing but container or vessels, like it's like a bucket which where we can but it will be in the large scale, and the big industries they will be having like wine factory or beer factory and all those things. These reactors is controlled environment will yield a useful substance in large amount, like that's what it is, not for the small amount. And it in controlled environment in the sense we are going to control everything like temperature, pH, pressure and all those things, so that the particular ingredient, the initial product which we have added, that can go to the final products into the larger quantity. Coming to the cell fusion also, the biotechnology techniques only. In these techniques involves the fusion of two cells to make a single cell that contains all the genetic material of the original cell. That's what fusion means. One cell and another cell, they will be joined together, and they are going to have the one another cell, new cell, which will be having the characteristic of the old cell. So far these techniques has been employed or used to create new plants by fusion cells from the species they don't naturally hybridize, right? So that's what two best characteristic of the plant. It may be the mango plant or it may be any medicinal plant. From there we are going to fuse the cell and we are going to get the good quality of the plant cell. Liposome based delivery. Liposome is nothing but these are the microscopic spherical structure that develop when lipids form a suspension in water, right? That's what it's a small what you call round shape, particular shape that will be having a lipid coat, and where we can load the drug, and it can be useful for the proper delivery. These spherical vesicles arrange themselves so as to generate a teeny space, right? Small space in the middle inside the center of the liposome, so that we can load the drug into this teeny space, and that can be that's what is microscopical spherical structure. Why it is told, it is very small and it can be inserted at any area of the body where it can deliver the drug. Such space can be potentially be exploited to deliver or transport the another substance such as a drug, right? So that is happening here in the case of liposome delivery system. Coming to the cell or tissue culture, these techniques allow the growth and a division of individual cells in a bath of sterile or nutritive fluid, which often contains hormones and growth substances, right? So what is happening here, it is allowing the growth and division of the individual cell, right? Cell culture or tissue culture, that's what, right? The providing that we are going to provide the proper hormones or proper growth. In the case of plant, we are going to provide them 16 essential elements and the non-essential element, macro element, micro element, nitrogen source, carbon source, all those things. If it will be provided, it can grow themselves in vitro or into the laboratory. This method is used extensively into the biological laboratory. That's what I told you. For example, in cancer research, for to study after isolation of the cancer cell from to the patient body, and it can be studied that what kind of pattern they are adopting to grow or uncontrolled growth, plant breeding for the getting the better quality of the plant. Two best characteristic of the plant cell can be cultured into the laboratory, and the routine analysis of the chromosome, like the karyotopes, right? So this can be done. Another thing, genetic engineering also, it is one of the technique which will be used into the biotechnology. The basis of genetic engineering is the alteration of genetic material like DNA, RNA, nucleic acid, like that's what hereditary material, that is also known as, or the combination of the genes in an organism, right? So that we can get the better quality or of hybrid quality of the particular organisms, or maybe it may be useful into the treatment, right? So that the hereditary disease like diabetes, hypertension, migraine, and all those things can be avoided. By modifying the organism, the genetic researchers give the organism and its descendant different traits, right? So it will be having different types of pattern or trait, so that it will be having different types of behavior. This technology was practiced in earlier times by breeding plants or breeding animals to produce the favorable combination of the genes, so that we can get the hybrid quality. DNA fingerprinting. DNA fingerprinting is the technique that is employed for identifying the components of DNA, right? So the material of the genes, that DNA, RNA, or nucleic acid, I told you, genetic material that are unique to the particular individual. That's what in the whatever whoever the person existing into the earth, they will be having different DNA. No DNA it is matching with each other. It is unique or to particular, so that will be useful for the identifying of the components, right? That's what it is used into the forensic lab and used into the different types of identification. Variation in DNA among different individuals can be used for identification purpose. That's what, right? In the crime scene, in the crime, whoever will be done any, so they will be collecting the biological material and they are going to find out the DNA, and they will correlate with the particular criminal and all those things. This small section of the DNA of an organism uniquely distinguishes or differentiate that the particular organism from all other, like whether it is the monkey DNA or rabbit DNA or rat or mice, or what kind of DNA it is, that can be identified by the proper sequences. Such varying bits of genetic material take the form of sequences of DNA called mini satellites, which are repeated several times, right? The next important technique, that is cloning. This method of production of the identical animals or plants or microorganism from a single individual, it is known as a cloning, right? So that's what cloning, it will be done like similar types of organism can be produced by the taking their genes and after cloning them, which will be having the same characteristic and features of the previous organism or parent organism. In the other word, we can say it is the process by which an organism is derived from a single parent through non-sexual production. Here we are not going to have the sexual production, right? Because it will be done in vitro or into the laboratory. The cloning has to be done, right? Cloning is the gifted in nature to those organisms that reproduce asexually and produce their own clones. Let's say for example plants, microorganisms, and a simple animals such as corals, right? So this will be done by this thing. Artificial insemination and the ET technology. What is the insemination? The development of in the study of embryology, urology, and urogenitology has led to progress in this area dealing with the artificial insemination. Here what we do is, it allows the artificial introduction of the semen. We need to understand here, semen into the reproductive tract of the female animal. Okay, so what will happen, it is extensively used in the breeding of the animal such as sheep and the cattle. So what will happen, no need to have their reproduction, only semen will be replaced into the uterus, and the fetus will develop, and the particular female animal, it will have the child, and it will be having all the characteristic feature of the particular male sheep or cattle. Male with the dominant or desirable hereditary trait or characteristic are selected for the semen collection. So whichever the breed or brand we are going to have, according to that we are going to select the semen, and that we are going to replace into the uterus of the particular female animal. Collected semen from the males and a desirable trait can be frozen and transported long distance from the fertile to fertilize the female animal. Like say for example a camel, it will be very good quality into the Saudi Arabia or UAE. So from there the camel semen will be collected and it will be introduced into the Indian camel. So whatever the Indian camel female it will have the baby, it will be expected to have the same kind of camel which is there into the Saudi Arabia, so that they can load over luggage and they can have the strong breed like that. Stem cell technology also, it is a very important and popular nowadays in the field of biotechnology. With the advancement of the biotechnology, it is now possible to utilize the potential of stem cells for the beneficial purpose, right? That's what it is nowadays. It is useful for the stem cell therapy. That's very good example. Stem cells are the undifferentiated and can the mitotically divide to create the mature functional cells. Say for example, bone marrow stem cells can give rise to entire range of the immune system blood cell, because we know that bone marrow, it is producing the T cell, B cell, and all other plasma cell for the immunity. So if it will be replaced or interfered properly, it can be, we can increase the immunity of the particular person or particular animal. Stem cells are found into the most organism, but they usually found into the multicellular organism, not into the acellular or unicellular. Generally it will be seen into the multicellular organism, say for rat, mice, human being, and all those things. In 1908, Alexander the Maximov coined or introduced the terms of stem cells, and later stem cell research work was continued by the Canadian scientist Ernest James in the 1960s. So it is still going on, right? So these are the different techniques which is you are using into the field of biotechnology. Thank you.